understanding the types of stamens in the floral morphology. So as I said, androecium, which we have understood separately, and how anthers and filaments have the cohesion, the different names for the same we have covered. Now in this section we can say about stamens. So there are few important things that we would understand here. First of all, the stamens can be equal or unequal in length. Let's say there are two long and two short stamen. This condition is known as Didynamous. A good example is Ocimum, right? The next is a case where there are four long inner whorls and two short outer whorls, right? And where does this occur? This condition is known as Tetradynamous, and this occurs in mustard. Clear? So that's one of the basic things. Now coming on to the dehiscence of anther, that means the anther when exposes the pollen grain, how it is done. It could be longitudinal, right? So when I say longitudinal, this longitudinal again can be of two types. This is known as introse and extruse. Longitudinal means a silt appears along the longitude. A good example is mustard. Now this longitudinal, as I said, could be introse or extruse. Introse is a silt towards the inner side of the or the center of the flower. Extruse is the slit towards the outer side of the flower. So it is towards the outer side and introse is towards the inner side. So where is the slit actually? Not the silt, it's the slit. So where is the slit actually is the important thing. So longitudinal is longitudinal in nature. Transverse is breadth-wise. So it is horizontal in nature. And this, a good example is malva. The next is porous. Porous is where on the tip there are small pores that occur. So we call it as solanum, right? Valvular is where the splits occur at several places, and that occurs by lifting the surface layers. So when you are lifting the surface layers and split occurs on various levels, it is known as valvular. Barberry is a good example for it. And then finally another one is irregular. A good example of irregular is najas. So those are the examples across each of the categories when we talk about the dehiscence of anther. So how anther actually has the dehiscence? It is either longitudinal, it is on the top at the pore, it is transverse breadth-wise, it is at the pore of the tip, or it is split by lifting the surface layer. So those are some of the things that we understand under stamen and the basics of stamen.